Hello students, welcome again to the channel Physiology Redefined. Today I will discuss the topics Neuromuscular Transmission. In my earlier video, I explained the structure of neuromuscular junction. So in that video, I mentioned that neuromuscular junction is the junction between nerve, that is motor neuron, and the muscle, which is comprised of the axon terminal, that is a knob-like structure, which is known as synaptic knob of the presynaptic neuron. Then there is a sarcolemma muscle membrane, and in between these two structures there is a gap containing extracellular fluid, which is known as the synaptic cleft. So these are the three components that I talked about while discussing the structure of neuromuscular junction. Now if a stimulus is given on the motor neuron, what's happened? The muscle which is supplied by that particular motor neuron, so this muscle will now contract, will give the response, and thereby our voluntary or involuntary activity of the muscle is getting initiated or is getting executed. So let us see in this video, in response to stimulation of the motor neuron, either by the external stimulus or the internal stimulus, how that electrical impulse or electrical signal is passed to the muscle, and thereby muscle contraction takes place. So in this video, I will try to explain how the electrical impulse is passed from the presynaptic neuron to the post-synaptic membrane, that is the sarcolemma of the muscle. And that process is known as the neuromuscular transmission. Transmission of the electrical impulse from the neuron to the muscle via the neuromuscular junction. That is the topics of discussion today. So please watch this video till the end. So now suppose this is the neuromuscular junction structure. This is the neuromuscular junction structure. It is the presynaptic neuron axon terminal of the presynaptic neuron containing the synaptic vesicle which contains the neurotransmitters. And this is the presynaptic membrane of the axon terminal. This is the synaptic cleft. This is the gap which is known as synaptic cleft. And this is the muscle membrane. This is the muscle membrane. Suppose this is the muscle membrane. So this is the structure which is known as the neuromuscular junction. Now here we will see how the impulse from this neuron is passed to the muscle. And that process is known as neuromuscular transmission. In the resting condition, let us see what's happened. And once the stimulus is given in the motor neuron, then what's happened. So in the resting neuron, like that of all the resting cells in our body, the inside of the membrane is electronegative, that you already know, and the outside of the membrane is electropositive. This is already known to you. So in the resting condition, inside is electronegative and outside is electropositive. Now what's happened? In the presynaptic membrane, what's happened? There are some channels, some channel proteins which are known as the calcium channel protein. So this is the calcium channel protein. These are the calcium channels proteins, or voltage gated voltage gated calcium channels, which remain closed at the time. So here these channels used to close at that time. Now what's happened if a stimulus is given? Suppose a stimulus is applied here. Stimulus is given in the motor neuron. A stimulus is given. As the stimulus is given, obviously what's happened? What will be the status of the membrane? What will be the status of the potential difference across the membrane? That is known to you. Yes, it will be the depolarization. So that membrane, presynaptic membrane, will get depolarized. That means inside will be electropositive, and outside will be electronegative. Outside will be electronegative. So this will be the state of the condition of the membrane potential across the presynaptic membrane in response to stimulus. So once the stimulus is given, action potential is generated here, is generated here, and that action potential will pass through the presynaptic membrane. And the action potential will pass means inside of the membrane will be electropositive in respect to outside of the membrane. So what's happened? Inside will be electropositive. That means a change of the voltage across this membrane takes place. Previously during resting condition it was electronegative. Now it has become electropositive. So change of the potential difference across the membrane takes place. And this change of the potential difference across the membrane from inside electronegative to inside electropositive, what's happened? Here you see the voltage gated calcium channels will open. So here you see there is a opening of voltage gated calcium channels. Calcium channels will now open. These channels are sensitive to change of the voltage. So named as voltage gated calcium channels. So these channels is now opened, which is present across the membrane, across the presynaptic membrane, that is to open. So as it is open and as the calcium concentration in the extracellular fluid that is present in the synaptic cleft. This is the synaptic cleft that I mentioned. So in the synaptic cleft, there is a extracellular fluid which has got more calcium concentration in respect to inside of these synaptic vesicles, inside of this membrane, inside of this presynaptic membrane. So what's happened? As the concentration gradient is there, calcium ion will now enter inside the synaptic vesicle, exoplasm of synaptic vesicles. So now, as the calcium channels are open and as the calcium concentration is high in the ECF, present in the synaptic cleft, calcium ion will enter inside the exoplasm or cytoplasms of the synaptic terminals. Now what's happened? These calcium ions will initiate some changes within this structure. Some changes will be initiated. So what changes? There is two changes will be initiated by the calcium ions. Entry of the calcium ions. What changes? First one is known as the electrical depolarization. So this calcium ion will initiate two changes. First one is the electrical depolarization. So here you see generally what's happened, the inside of in the resting condition, once the stimulus is not applied, I told the inside of the membrane is electronegative. But the synaptic vesicular membrane, outside of the synaptic vesicular membrane, is also electronegative. So outside of the synaptic vesicular membrane is electronegative, and inside of the presynaptic membrane is electronegative. So there is a electrostatic repulsion between the synaptic vesicle and the presynaptic membrane. There is a electrostatic repulsion between the synaptic vesicle and the presynaptic membrane. So synaptic vesicle will not come at the linings of the presynaptic membrane. Synaptic vesicles will not lie on the presynaptic membrane because of the electrical repulsion between the presynaptic membrane and the synaptic vesicular membrane. Now, as here calcium ions are coming in, as the electrical depolarization has taken place, as the electrical repulsion has taken place, electrical depolarization has taken place, what's happened? Now it is positive, and outside of the membrane is electronegative. So inside of the membrane is electropositive, and here it is electronegative. So inside of the membrane is electropositive, and here it is electronegative. So now opposite charges are there. As the opposite charges are there, now synaptic vesicles will be attracted towards the presynaptic membrane, interior of the presynaptic membrane. Synaptic vesicles will now be attracted towards the interior of the inner aspect of the presynaptic membrane. This is number one. So calcium ion will initiate these changes, electrical depolarizations, which results the attraction of synaptic vesicle towards presynaptic membrane. This is number one. Number two, what's happened? This calcium ion once enters inside the exoplasms of the synaptic terminal. Then what's happened? This used to phosphorylate some proteins which actually act as a glue and thereby help the synaptic vesicle to attach on the cytoskeleton. So this cytoskeleton, the microfilaments, you know, these are the microfilaments. So these on the microfilaments, synaptic vesicles are normally attached. So in the cytoskeletons of the synaptic terminal, synaptic vesicles are bound. They do not allow them to fall towards the presynaptic membrane because of some proteins which are acting as a glue, which are acting as a adhesive material that actually used to bind the synaptic vesicles with the microfilaments. So they do not fall towards these things. Now what's happened? Once the calcium ion comes in, this will phosphorylate that protein. So phosphorylate the protein, and this protein as phosphorylation of the protein is there, no binding of synaptic vesicle with microfilaments, and thereby both these phenomena, on the one hand, synaptic vesicles are no longer bound with the cytoskeletal structures, bound with the microfilaments. This is number one. So they are now detached. Synaptic vesicles are now detached from the cytoskeleton. On the other hand, there is a electrical attraction. There is an attraction of the synaptic vesicle towards the interior of the presynaptic membrane. And this is because of these two things, two changes, because of the calcium entry, the synaptic vesicles will lie on the interior or inside of the pre-synaptic vesicles. Here you see synaptic vesicles are lying on the pre-synaptic membrane, on the pre-synaptic membrane. It is no longer present at the cytoskeleton, at the exoplasm. They are lined on the pre-synaptic membrane. Now this attachment of the synaptic vesicle on the presynaptic membrane, this phenomena is known as docking. So this phenomena is known as the docking. So this is the phenomena which is known as the docking. So what's happened in this docking? What is actually docking? Docking takes place. Here you see, suppose this is the synaptic vesicle which contains the neurotransmitters. Now on the wall of the synaptic vesicles, there are some proteins which are known as synaptobrevin. So on the walls of these things, there are some protein which is known as synaptobrevin. And on the presynaptic membrane, this is the suppose presynaptic membrane. This is the presynaptic membrane. So in the presynaptic membrane, on the other hand, another type of protein is there which is known as Syntaxin. So this is known as the Syntaxin. So this is the protein known as Syntaxin, and this is the protein which is known as synaptobrevin. So this synaptobrevin protein gets locked with the syntaxin, and this interaction between the synaptobrevin and the syntaxin is also helped by a lot of proteins present at this part, and those proteins are collectively known as the SNAP proteins. So besides this synaptobrevin and syntaxin, there are different type of other proteins which are known as the SNAP proteins. So in association with the SNAP proteins, syntaxin and the synaptobrevin gets locked, and thereby the synaptic vesicle get fixed on the membrane, pre-synaptic membrane, and that is the process, that is the phenomena which is known as the docking. So this phenomena is known as the docking, and docking takes place because of the interaction between two proteins, one present in the synaptic vesicular membrane known as synaptobrevin, and another present on presynaptic membrane that is known as syntaxin. Their interaction is assisted by a lot of other proteins which are known as the SNAP proteins. Different types of SNAP proteins are there. So this process is known as the docking. Now once the docking is done, the synaptic vesicles are fixed or locked on the presynaptic membrane. Then those calcium ions also initiate some other chemical changes, activation of different enzymes, and this activation of the different enzymes ultimately helps the release of the neurotransmitters from the synaptic vesicle, and that phenomena is known as priming. So docking and the priming. So docking is the attachment between the synaptic vesicle and the presynaptic membrane, whereas priming is the activation of the synaptic vesicle to allow the release of neurotransmitters from the synaptic vesicle. So that activation process is also initiated by the calcium ion, which is known as the priming. So ultimately what's happened, here you see there is a fusion of the synaptic vesicular membrane and the pre-synaptic membrane. Synaptic vesicular membrane gets fused with the pre-synaptic membrane, and thereby neurotransmitters which were present on the synaptic vesicles are now exposed to the extracellular fluid present in the synaptic cleft. That means there is a release of the neurotransmitters, and this is the process which is known as the exocytosis. So by the process of exocytosis, fusion of the synaptic vesicular membrane takes place with the pre-synaptic membrane, and thereby neurotransmitters are released in the extracellular fluid. Now, these neurotransmitters once get released at the synaptic cleft where ACh is present. What's happened? These neurotransmitters, here you see, these are the neurotransmitters. These neurotransmitters is known as the acetylcholine. So this is the neurotransmitter which is known as the acetylcholine. So this acetylcholine will now gets bind with the receptors, present on the muscle membrane. So this is the muscle membrane. So on the muscle membrane, there are some receptors which are known as the ligand-gated receptors. These are the ligand-gated receptors. This has got the ability to bind with the chemicals. So there is a receptors, and these receptors are known as the nicotinic acetylcholine receptor. So once the acetylcholine neurotransmitter binds with the nicotinic acetylcholine receptor, then what's happened? These receptors are coupled with, they are present in association with some channel proteins which are known as, these are the channel proteins which are known as the ligand-gated ion channels. Generally, the sodium channels, ligand-gated sodium channels are there, which used to close once the acetylcholine is not bounded with the receptor. In the resting condition, once the acetylcholine is not released, then these ligand-gated sodium channels used to close. So once the neurotransmitter gets bind with this, here you see, once the neurotransmitter gets bind with the nicotinic acetylcholine receptor, there is an opening of the full ligand-gated sodium channels. These sodium channels will now open. Here you see, there is an opening of the ligand-gated sodium channels. So opening of ligand-gated sodium channels. Here I told the opening of voltage-gated calcium channels. Here it is the ligand-gated sodium channels. They used to open. And as the sodium channels is now open, and as the sodium concentration is high in the outside, in the ACh, this sodium ion will enter inside the sarcolemma, inside the muscle. And as the sodium ions are coming in, what's happened? Change of the membrane potential of the sarcolemma or muscle membrane. Normally, in the resting condition, it is minus 90 millivolt. So now what's happened? Sodium ions are coming in. As the sodium ions are coming in, as the positive ions are coming in, inside electronegativity will be now decreased. And this inside electronegativity is decreased to minus 70 millivolt. So because of the sodium entry, inside electronegativity of the muscle membrane will be decreased to minus 70 millivolt, and this is the potential change that is known as the end plate potential, EPP, end plate potential. So once the end plate potential is produced, that means from the resting condition, that is minus 90 millivolt, the inside electronegativity falls to minus 70 millivolt. When from minus 90 it becomes minus 70. So this change is the EPP, end plate potential, from minus 90 to minus 70. This change is known as the end plate potential. So once the end plate potential is developed, then what's happened? There is a opening of voltage gated sodium channels present on the muscle membrane. So they act as a firing level. This act as a firing level. And because of this firing level, what's happened? Because of the firing level, the action potential is initiated. Action potential is initiated on the muscle. So this muscle will become now electropositive. Inside will be electropositive in respect to outside of the membrane. So in the resting condition it was inside electronegative. Now what's happened? It will be inside electropositive. And inside electropositive because of the entry of the sodium ions. And this is otherwise known as the depolarizations of the sarcolemma or the muscle membrane. Muscle membrane is depolarized, and that is the first phase of the action potential. So in this way, the action potential that is initiated in the motor neuron, that is passed to the muscle membrane. So action potential or electrical signal which was generated at the motor neuron, that is passed to the muscle membrane via the release of calcium, via the release of neurotransmitters from the synaptic vesicle. And this is the mechanism, this is the process by which the electrical signal is passed from pre-synaptic neuron to the post-synaptic neuron. So from the pre-synaptic neuron to post-synaptic neuron, the electrical impulse is passed. And this is the process which is known as the neuromuscular transmission. Now, hope this basic mechanism is very clear to you. Now, one thing I want to mention, that as I told that end plate potential, is reached from minus 90 to minus 70 millivolt. And this minus 70 millivolt, to produce the minus 70 millivolt end plate potential, more than sufficient amount of the acetylcholines are released from the presynaptic terminal. As I told, that EPP is developed because of the sodium entry. Sodium entry is because of the binding of the acetylcholine on the receptor, and this binding of the acetylcholine is due to the release of the acetylcholine. That means the acetylcholine neurotransmitter has to be released to produce the end plate potential. Now, to produce the end plate potential from minus 90 to minus 70, whatever amount of acetylcholines are required, around 10 times more than amount of the acetylcholine are released in response to its stimulation. That means, suppose to produce the end plate potential on the sarcolemmal membrane, suppose the 10 molecules of acetylcholines are required, suppose. But in reality, what's happened? 10 into 10, 10 times. So 10 into 10, that means 100 acetylcholine will be released. because of the stimulation, so that there is a no failure of initiation of the muscle contraction in response to acetylcholine or neurotransmitter release. So to avoid any failure to generate the action potential on the muscle, to depolarize the muscle, around 10 times release of the acetylcholine takes place, and that is the factor, is known as the safety factor 10. Safety factor 10. So this is the factor which is known as safety factor 10. That means 10 times release of the acetylcholine from the synaptic terminal than the requirement to produce the endpoint potential. So this is the factor that is known as safety factor 10. Now another thing, as I told, that acetylcholines are released by the process of exocytosis. Acetylcholines, neurotransmitters, are produced. Now these acetylcholine neurotransmitters are not produced as a continuous water flow. It is not like a continuous flow. It was the pulsatile release. Switched on, switched off, switched on, switched off, switched on, switched off. There is a pulsatile release of the acetylcholines, and this quantal release of acetylcholine is known as the Dale's phenomena. So this acetylcholine release is the quantal release. And at a time, what's happened? Around 60 to 200 vesicles are released. At a time, 60 to 200 vesicles are released. 200 vesicles are releases their secretory substances, that is acetylcholine, and this is the phenomena which is known as the Dale's phenomena. Now, related with this neuromuscular transmission, there are some diseases. One of the very important disease is known as Myosthenia Gravis. So what's happened? As I told here, you see here that this is the voltage gated calcium channel. As I told, voltage gated calcium channels. Now, if these channels are abnormal, that happens in case of some development of antibodies which used to destroy these voltage gated calcium channels. So once, if within the body, due to any reason, some autoimmunity develops towards the voltage gated calcium channel proteins, then what's happened? These voltage gated calcium channel proteins will not work. So they will not open. As they will not open, no question of calcium entry. So no question of docking and priming. So no question of acetylcholine release, and thereby no question of the stimulation of the muscle. And this abnormality, this disease is known as Lambert-Eaton syndrome. Lambert-Eaton syndrome. So Lambert-Eaton syndrome is the weakness of the muscle, gradual weakness of the muscle, followed by the failure of the muscle, permanent failure even to the muscle, or muscle paralysis. And this is due to the destructions of the voltage gated calcium channels. Voltage gated calcium channels are damaged by the antibodies developed against it, and that disease is known as the Lambert-Eaton syndrome. Another type of same type of disease takes place once the same different other type of antibodies are developed against the acetylcholine receptor. As I told here, this is the acetylcholine receptor. This is to bind the neurotransmitter acetylcholine, and this binding initiates the opening of ligand-gated sodium channels, and thereby sodium enters to develop the endpoint potential and thereby action potential. So now happen, if the antibodies are developed against this nicotinic acetylcholine receptor, if these antibodies are developed against this nicotinic acetylcholine receptor, then what happens? These acetylcholine receptors will get damaged. So if these are damaged, acetylcholine, even if it is released, they cannot bind with the receptor, and thereby no questions of sodium entry, no question of the EPP formation, no question of action potential formation, and thereby no question of excitations of the muscle, so no contraction. So same complication will take place, but the cause is different. Here cause is damage of the nicotinic acetylcholine receptor, and this is the disease which is known as the Myosthenia gravis. Myosthenia gravis. So two types of disease can take place due to the autoimmunity, due to the antibody formation. One is antibody for against the voltage-gated calcium channels. Then that disease is known as Lambert-Eaton syndrome. And if the antibodies are formed against the nicotinic acetylcholine receptor, then that disease is known as myasthenia gravis. In both the cases, complication is same, only the cause is different. So one is the Lambert-Eaton syndrome, another is the myasthenia gravis. Now another thing I want to add in this video, that there are different types of neuromuscular blocker. One type of neurotransmitter blocker, one type of neuromuscular blocker is known as the pre-synaptic blocker. Pre-synaptic means if the blockage takes place at the pre-synaptic membrane, that means acetylcholine is not released, neurotransmitters are not released, then that type of some drugs which blocks the release of the acetylcholine, and then that type of blocker is known as the pre-synaptic blocker. Examples of the pre-synaptic blocker is the botulinum toxin and hemicolinium. Botulinum toxin. So what's happened? There is a drug known as the botulinum toxin. So what's happened? This botulinum toxin, they used to damage the proteins, either the synaptobrevin or syntaxin or SNAP proteins. So these toxins are responsible for damaging of the synaptobrevin present on the synaptic vesicular membrane, or syntaxin present on the presynaptic membrane, or SNAP proteins. Then what's happened? There is no docking of the synaptic vesicle on the presynaptic membrane, and as the docking doesn't take place, no priming and thereby no exocytosis, so acetylcholine will not release, and thereby there is a blockade of the muscle, and thereby muscle will not contract, so they will remain in the relaxed state. So this is the blocker that is known as pre-synaptic blocker. So this is the pre-synaptic blocker. Pre-synaptic blocker. Pre-synaptic blocker. Second type of blocker, neurotransmitter blocker, is known as the post-synaptic membrane. Post-synaptic blocker. That means here Here blocking is taking place at this post-synaptic membrane. Now this post-synaptic membrane can be blocked by different ways. Some blockers are there which are known as the competitive inhibitor. They compete to the acetylcholine and thereby do not allow the acetylcholine to get bind with the acetylcholine receptor. So they are the competitive inhibitor which competes with the acetylcholine. So they gets bind with the acetylcholine receptor and do not allow the acetylcholine to get bind with its receptor. As a result, no binding of acetylcholine with the receptor. So no questions of opening of ligand gated sodium channels and no question of depolarization of the muscle membrane and thereby no question of muscle contraction. Muscle will be in relaxed state. So this type of blocker is known as the post-synaptic blocker. So one type of post-synaptic blocker, so this is number one, pre-synaptic blocker. Second type is the post-synaptic blocker. Post-synaptic blocker. And these are number one is the competitive inhibitor. Competitive inhibitor that I explained. This is the curare and the galamine. Curare and example is the curare and galamine. So these are the two types of drugs which act as a competitive inhibitor. Second type of blocker is known as the depolarizing blocker. So what does it do? This depolarizing blocker. Now here one thing you see. As I told, acetylcholine binds with this and initiates the muscle contraction, muscle excitation followed by contractions. So once its action is over, these acetylcholines are destroyed or metabolized by an enzyme present on the basal lamina of this post-synaptic membrane. There is a basal lamina. So on the basal lamina, there is an enzyme which is known as the acetylcholine storage. So this acetylcholine storage metabolizes the acetylcholine. So acetylcholine will be exhausted. Now this depolarizing blocker, what they do? They do not allow the metabolism of the acetylcholine. So acetylcholine will be present for the long period. As it will be present in the long period, muscles will be the contractile. So even if the next stimulus is coming, muscle will not be relaxed. So muscles will remain in the contractile state and ultimately for the prolonged contractions, after prolonged contractions, it will be in the totally relaxed, totally exhausted and thereby in the relaxed state. So this is the blocker which is known as the depolarizing blocker, and examples are succinylcholine and carbamylcholine. Third category of the blocker, that is known as the acetylcholine storage inhibitor. Acetylcholine storage inhibitor. One is the reversible type, another is the irreversible type. In case of irreversible inhibitor, what's happened? The acetylcholine storage enzyme is getting inhibited. Now what's happened? This is getting inhibited, and as this is getting inhibited, what's happened? Again, there is a prolonged exposure of the acetylcholine with the receptor, and the same phenomena will take place as I discussed under depolarizing blocker. And this acetylcholine inhibitor can be of reversible type, can be irreversible type. in case of in case of reversible acetylcholine receptor, reversible acetylcholine receptors, examples are the tetrodotoxin and sagittauxin. So they can bind with this acetylcholine enzyme, inhibit them for the temporary period. And so this is the reversible blocker. And there is another type of blocker that is known as the irreversible. Examples are the organophosphorus compound, the nerve gas. So these are the different type of chemicals which permanently blocks the or inhibit the acetylcholine inhibitor, and thereby ultimately relaxes the muscles. So muscles will become flaccid, muscles will become paralyzed. And so these are the different two types of acetylcholine storage inhibitor. So all these are the examples of the post-synaptic blocker because they act at the post-synaptic membrane, that is the muscle membrane. So this is all about the neuromuscular transmission along with its applied aspect. I hope you will like this video. If it is liked by you, please share it to your friends, and obviously don't forget to subscribe this channel if not yet subscribed. Thank you.